Ladies and gentlemen, we're ready for the last distinguished speaker, Dr. Maneesh Paul, who will be talking about challenges in developing effective antimicrobials against XDR and PDR gram-negative bacilli. And to welcome him, we have distinguished moderator, Dr. Abdul Ghafoor. And to welcome him, we have another dashing PI from Team Dash, Dr. Rajni. Dr. Rajni, will you please introduce yourself and then do the honors?
Yes, ma'am. A very good evening to everyone. I'm Dr. Ekadashi Rajni. I'm a graduate from Lady Hardinge Medical College and a postgraduate from Maulana Azad Medical College, where I had the very good fortune of meeting Meher ma'am. She was my senior in Maulana Azad Medical College. And currently, I'm Professor Microbiology in Mahatma Gandhi Medical College, Jaipur, Rajasthan. So it's my proud privilege to introduce Dr. Abdul Ghafoor. And he is a senior consultant and adjunct professor in infectious diseases at the Apollo Hospitals, Chennai, India. Among several other achievements, Dr. Ghafoor is the primary author and coordinator of the Chennai Declaration. This is a renowned Indian document and initiative to tackle the challenge of antimicrobial resistance. He is a former technical advisory committee member for the National Antibiotic Policy, Ministry of Health, extremely active in academic, policy, research, socio-economic, and political aspects of infectious diseases. He is the managing partner and lead strategist at Quorum Veda Consulting Services. Again, then he is an advisory and jury member of the Longitude Prize, delivered several lectures, and has a series of prestigious international publications in journals like BMG, Lancet, etc. He is on the editorial board for several international journals and received the Indian Medical Association Oration Award for his contribution towards tackling antimicrobial resistance efforts. With this, I'm very happy to welcome Dr. Ghafoor on this online platform to moderate the next session. Please, Dr. Abdul Ghafoor.
Thank you, Dr. Rajni. Yeah. Our next star speaker is Dr. Maneesh Paul. And I'm not going to read out his TV slide, because if I do that, I will be diluting his most important achievement. That is, he is the lead co-inventor of the first Indian anti-infective to the world, that is cefepime and Enmetazobactam. Dr. Maneesh, I'm an infectious disease physician working in a cancer center. Every day, I treat patients where only 1 or 2 antibiotics are effective, or no antibiotics are effective. And when I'm holding their hand, I feel helpless. And for myself and for my patients, any scientist who develop, invent, and deploy new antibiotics is an angel and a godsend. So to me and my patients, Dr. Maneesh, you are an angel and godsend. Over to you. We are so enthusiastic to listen to your story. Thank you, Maneesh. Over to you.
Thank you, Dr. Ghafoor. Thank you, Dr. Meher, for the opportunity. Hey, I would like to have the privilege to share the presentation. I have it now. Thank you. And also, it is so wonderful to listen to Dr. Meher, who put up the complete background, so it saves a lot of time for us to repeat those things, and also endorsed by Dr. Neelam Taneja. She did give about brilliant data. One thing I would like to slightly say differently is, we always say India is the hub for AMR, but I don't think that's so true, because AMR is a global challenge. We have AMR all over the world. And Indian data, though we collect, such a massive country, right? It wouldn't definitely not represent the AMR of the entire nation per se, but only the collected data, of course. And of course, thanks to Dr. Livermore, and he has been one of our mentors during the invention of Enmetazobactam, in addition to many other scientists. And yes, thanks, Dr. Sardana, for moderating that quite well and concluding it in a very precise manner. And it's beautiful to listen to Dr. Nordman. The importance of diagnostics is so critical. Coming to my topic, challenges in XD, extremely drug-resistant gram-negative bacterial anti-infectives. This is a an area where I think all of us struggle a lot. Minimize this, okay. Without going too much detail into these facts, which everybody is aware of it, I would like to highlight the blue line here. AMR is complex and no individual nation can address this major public health problem alone. And it fits quite well with the topic of today's presentation and the whole symposia itself, that it is one health approach we have to take. Because anytime we say we are not looking at only one sector, or when we look at holistically, but not practically translate that, it just becomes a moot point. And of course, coming to the major area of multiple drug-resistant bacterial infections, now, even the last resort antibiotics, right? Like Dr. Nordman showed the data, it is really scary to see the way KPCs have also evolved. I say KPC 156 or 163, I think, and that's scary. We are going on identifying newer KPCs and newer carbapenemase producing bacteria. And when bacteria become so multiple drug resistant, the challenge comes is with the discovery of novel antibiotics. Because every time, as we know from the history of anti-infectives itself, when the first antibiotic, penicillin, came into existence, before it was approved, we had MRSAs in the circulation, which means to say that we have already wisdom of AMR among the microbes much long before we could even ever think of identifying or discovering and inventing any new molecule. So the way I say it is, every time we say, hooray, we got a new antibiotic, microbes would say, okay, we know about it. It's no big deal that you invented a new molecule because we know all about it. So we have every arsenal with us how to combat it. So this is where how our collective human intelligence, collective I say because it is not one person's activity, even inventing this molecule in Enmetazobactam, though I was the lead biologist there, it is not one man's activity. It is a collective activity of so many people in the company who worked quite hard. We had in vitro scientists, we had in vivo scientists, pharmacologists. It's a whole journey. I'll talk about it a little while. Now, what we have to do is we have to re-engineer the approaches of existing antibiotics. Inventing newer molecules is very good, but we already have exposed the bacteria to a certain set of antibiotics. Can we re-engineer them? Can we bring something supporting these antibiotics? So that's when we have to figure out how antibiotic against drug-resistant bacteria can be increased through structural modifications, right? And that's where we identified Enmetazobactam as well. So further to that, we have to understand the regulatory and translational challenges for antibiotic resistance. Because now when you're re-engineering any molecule, because bacteria know this, any modifications around it, then for a microbe to become resistant to that is quite easy. Oh, here is where we have to look at a different approach. And what is that approach would be? I prefer to look at the virulence factors of the microbes rather than going at the microbes directly. Because anything we go directly at the microbes, they would again reinvent themselves and reactivate themselves multiple ways. So instead of triggering that activity, if I can attack its virulence factors, one of them being the beta-lactamases, right? And that is the approach. What I'm mentioning here is Enmetazobactam, where we have approached attacking its enzymes rather than the bacteria itself. So here, in a way, bacteria doesn't know exactly, oh, there is a new BLI there. It is just confused now. Well, we have, again, Cephalosporin attacking us. What's going on? So they may be thinking that humans have surpassed them. But before they catch up, I think we can take care of the carbapenems. Because when we start using carbapenems as the first line of therapy, we are, in fact, pushing the microbes to the corner to bounce back at us. So to prevent that, we need newer BLIs, which would come. Of course, we have quite a few have come up, and I would appreciate even the Zainich, which is the Zidibactam from Wokhart. That's from India again, the second molecule, which from India, which has made it to the market now. Of course, that is the one which is now, it is not exactly a complete BLI. It has also inherent antibacterial activity, but it does supplement the antibiotics, whichever it can be attached with, so that carbapenem resistant microbes can also be treated with that without introducing high pressure by the carbapenems themselves. So going to the One Health side, we have heard quite a bit of it on One Health, what it is and why we have to collaborate between all the three verticals. Because if we do not work in tandem, I think we are going to lose the battle again. The food industry. Of course, Dr. Taneja said we shouldn't blame the food industry or the agriculture or animal sectors, but I think it is not to blame anyone. Each of us have to own up the responsibilities in our own verticals and come collectively. And as she mentioned, there is no coordination at the policy level of all the three verticals to come together and address this challenge. So that is something as a policy matter we have to address. Because when we do that, we are going to take up and address the 5 sustainable developmental goals, which are critical as part of the 7 SDGs for the world to become more sustainable. We can address poverty issues, health and well-being, inequalities, hunger issues, and also the loss of manpower, which could happen due to highly drug-resistant infectious diseases. Now, we always think, where exactly is the infection coming from? Though this is a little older paper, maybe about a decade old, but still it is quite relevant to us to look at how anti-infective resistances or antimicrobial resistance comes from multiple sectors. And when I see this, the right side circles, the animal and humans are quite equivalent. And they are the ones which are contributing to the high resistances. So food animals are one more area where we have to pay attention to. Now, when this connection happens between all the three, we can definitely look at how to address AMR. And I'm not going to go into details of this, but in addition to all those things that were mentioned, I would bring in one aspect. I'll skip this slide as well. Access to antibiotics. This is very important aspect of controlling AMR. This was not touched, so I deleted other slides. The next 2 slides, I'm going to talk about why it is important to look at access to antibiotics as well. If there is no access to the right kind of antibiotics in the clinical setup, the clinician is helpless. When a patient walks in, they have to deal with it. So what do? So improve access. GARDP, the Global Antibiotic R&D Partnership, an initiative from WHO, I think that is doing an amazing job. And cefiderocol is one of the molecules that is going to come into the market very soon, I believe. And they're looking at multiple programs to ensure that these antibiotics, which are not known to add additional antibiotic resistance mechanisms, can be brought into the market so that the LMICs, which is a major challenge, where the major challenge is access to antibiotics, can be addressed. And the next important thing is the quality of antibiotics. We have a lot of substandard and falsified antibiotics available in the market, nearly 40% of them. This is what the WHO report says. And this is yet another reason why we have a major challenge in addressing multi-drug resistant organisms. Because when substandard means when it has got lesser potency than the available potency, approved potency, or we all journey so many years to bring a molecule to the market, and then if the set dose, the therapeutic dose, is not maintained in the formulation available over the counter or when it is administered to the patient, microbes are going to develop resistance again. So quality of the antibiotics is something very, very, very important aspect we have to pay attention to. Now, what are the major challenges? One of the things is the vulnerability of antibacterial drug R&D, because whenever we talk about antibiotic resistances or drug development, the major thing people say is, oh, there is not many molecules, not many avenues. That's not true. We have done so many research. I think, Dr. Ghafoor has started a group called Entrepreneurship Group, Innovation Entrepreneurship Group, wherein he's trying to bring in clinicians to be part of becoming innovators or entrepreneurs themselves. That is a very good initiative to engage them to understand the problem, because they know the reality in the clinics. And if they can partner or themselves come up with either diagnostics or finding newer ways of treating the infectious diseases, I think I'm looking specifically for infectious diseases, though that is broad. His forum is broad one, but infectious diseases, I think that is going to address quite a lot of problems. So some of the highlights I mentioned is the critical medical needs will be addressed by and understood by them, but the initiatives, right? Push and pull initiatives have been done by the global agencies, but it is not yet fully implemented. Only thing I can look at is the CARB-X, the BARDA, those things have come into play. But the initiatives where once the product comes to the market, how could we push it to reach the clinicians? Because drug discovery journey of anti-infectives is a very, very, very arduous journey with low return on investment. That is the reason there are very few companies who are actively working on it. That's when push and pull incentives become highly, highly important. And then we have to look at also non-traditional approaches. I will touch a slide upon that later. Now, here what we have to do is we have to look at the impact on bacterial growth and also virulence indirectly, as I mentioned earlier, because when we look at these indirect methods, I think we can address them as well. This is, we are conditioned to think that I have to hit the molecule directly, only then I can kill the microbe. No, there are multiple ways, just like the BLIs. When you look at the recent studies and phase 3, predominantly most of them are BLIs. Out of 28 small molecules in clinical trials, 21, I mean, most of them are BLIs, right? So, which is a good approach in my opinion, but of course we need newer molecules as well there. Now, what are the things we look at? The efficacy, the safety, right? These two elements are very important because many of the molecules which are in vitro active do not translate into in vivo efficacy. So that is where again the iteration and the loss of molecules is significantly higher. Then I just put the slide here. I'm not going to read everything and mention everything. Probably people would see it later. And I know it has been streamed online. People can always go back to watch the video to look at these slides again. The other point which was mentioned by Dr. Meher as well is about the impact it has on the microbiota. Right? How our gut microbiota forms the major, our human microbiota forms a major part of our human body's population of microbes. And we are specifically when we target the bacteria which are infecting, we may be affecting even other healthy bacteria. That is something we have to pay attention to. Now, we have to balance between refinement and innovation. I say this because when you're inventing me too drugs, when they come into play, they become resistant quite faster. That's when you're doing me too, go after BLIs, not after the novel molecules. That is one mantra, right? So the degree of innovation of new antibiotics definitely varies significantly because we have to look at same target, different structure. That is when we don't have to discover newer targets so much. Of course, we do need them, but existing to make it faster when the pipeline is so depleted, go to the same target, but come up with a different structure which the bacteria cannot easily destroy. Then look at different binding sites. The same, that is called repurposing. We have to repurpose many things. For example, we have the oxazolidinones, right? Which is a MAO inhibitor now, which is coming to antibacterial use. Similarly, look at the same molecule. If you identify newer targets, see whether it can bind to that site. Then we look at same pathway and different target and a newer target with the new pathways. These are these several ways how we can look at innovation of novel antimicrobials. This is a fundamental slide. All of us know about it. But this is where I want to tell you the arduous journey. When you identify the disease, coming to a target, it takes a long journey. Then we have hits identified by designed and developed by the medicinal chemist, which we have to find activity. The in vitro biologists work very hard to find that hit molecule. We had great number of team members in ORCID. We had in my team, the one, Mr. Shakti Singh, Solanki, we had Bharani, who were quite a hardworking people. They worked quite hard to identify novel molecules. Then it went to in vivo, where we had Hariharan, who is now with Wokhart, and he's done amazing work in discovering and developing Enmetazobactam. Then we come to clinical candidates, then drug candidates, and then comes the regulatory. These are the higher challenges. And when developing an anti-infective molecule, this is the journey I was talking about. Look at every stage, the failure rate is very, very high. The medicinal chemist makes the molecule. Look at the date, stage 1 is 2006. And when we finished out-licensing, it was 2013. And it came to market only in 2024. So that is the length of the journey. And we have to look at its MIC at the in vitro level, look at the broader set of organisms, look at the multi-drug resistant organisms, then bring in the efficacy activity, look at the tissue availability at the organ-specific regions by using pharmacodynamic models, such as thigh infection model, lung infection model. Then we come to look at multiple strains, how many strains are available within India, and look at those strains. That's what we did. Then we go for safety. Finally, we come to validation of a candidate. This is the journey of inventing a new molecule. And once we did that, we look, and this triangle is very important to identify our therapeutic window, because we have to balance between toxicity and efficacy and to make sure we hit the molecule at the same time do not cause any toxicity. And PKPD drivers. Normally, when you look at, we look at pharmacodynamics of the host and the drug. But in anti-infectives, I always put the third eye and say it is a microbial dynamics, because microbes can break the molecules as well and also alter the host as well to a large extent. So looking at microbial angle is very important when you're discovering antibiotic. I'm almost at the end of my slides. So this molecule was invented by Orchid Pharma, right? And up to phase 1 ready, and we out-licensed it to Allegra Therapeutics. And right now I'm driving the phase 4 clinical trials for this in India. And in parallel, the drug has been launched in India on October 1st. So I'm proud to say that I thank my entire team who worked with me across the spectrum, not to mention any names. Across the spectrum, people have worked very rigorously to bring this molecule to the world. And Allegra Therapeutics has done an amazing job in clinical trials and getting approvals from the FDA, the EMA, and the UK MHRA. And India has worked very well as Orchid Pharma to get our approval for the Indian regulator. These are a few of the alternatives. I just keep the slide there and I move to the next one, which are conclusion slides, right? I would go to this slide mainly for the want of time. We have to, the last one, we have to work together to end this AMR crisis. Policymakers, drug discovery companies, clinicians, and above all, I would say the citizens of the world should come together, because AMR can be addressed only by engaging every person on this earth. If we become responsible in the way we use the antibiotics, I think we have solved 99% of the problem. We don't have to worry much. And that is where I would encourage, stewardship as a major contributor and contributor in addressing AMR rather than only drug discovery and putting the onus on the clinicians or the pharmacists. Regulations have to come in, but if citizens become responsible, I think we have solved half the problem. Thank you very much.